Welcome back to The Dish. My name is Johannes, founder and CEO at LabCentral, and I'm excited to welcome today Noor Jailkhani, the founder and CEO of one of our startups, Matrisome. Noor, in order for the audience to get to know you and the company, could you get started by giving us a 30-second elevator pitch?
So my name is Noor Jailkhani. I am the co-founder and CEO of Matrisome Bio. Metastatic solid tumors represent a huge unmet clinical need, and they highlight the current limitations of cancer therapies that target cancer cells alone. Matrisome Bio is a targeted therapy platform where we're developing novel medicines for the treatment of advanced stage metastatic cancer by leveraging a new class of targets called the extracellular matrix, or collectively the matrisome. The matrisome is essentially a network of proteins that is outside or surrounds every cell of the body, including cells of tumors and metastases. Our technology converts this matrisome from a barrier into a drug depot. Matrisome's platform is both cancer and payload agnostic, and it is based on decades of research and IP that was built at MIT. Our diverse team of founders, investors, and internal team are really focused on developing first-in-class and best-in-class matrix-targeted therapies to support our mission of improving the lives of patients with advanced stage metastatic disease.
Excellent. Thank you. And we will get into a lot more detail about how Matrisome does that and the science. But before we do that, who is Noor? How do you find yourself as the CEO of an MIT spin-out startup?
So I'm essentially a scientist turned entrepreneur, first-time founder, first-time CEO. And I think my journey has really been fueled by a deep interest in life sciences and biomedical research. For as long back as I can remember, I've been interested in biology. I grew up in India, in New Delhi, on the campus of actually a medical school. Both my parents were professors of biochemistry, so they had their labs and they did their teaching. So in some ways I was influenced by my environment, and I think that interest in biology took me to an undergrad in microbiology and a PhD in immunology.
Did you do both of these trainings in India?
I did. My entire educational training happened in India.
Okay.
All the way up to my PhD. Mm-hmm. And then once I finished my PhD, there were personal events that allowed me to get access, or I'd say exposure to the dire needs of patients with advanced stage metastasis. Mm-hmm. And so I pivoted to oncology. And I thought about what is the best place to do cancer research. And that is what brought me to Boston. And when I came to Boston, I was really fortunate to come across the Koch Institute for Cancer Research, where I trained in the lab of Richard Hines, which had been studying tumor progression and metastasis, and specifically tumor microenvironments. And a big component of that, which is the extracellular matrix, collectively the matrisome. So I was a trainee in Richard's lab, postdoc for 5 years, then I stayed on as a research scientist. I had never imagined that the science and the research that I do in his lab would then be the foundation of Matrisome Bio.
Wonderful, that's great. Did your parents have any entrepreneurial activities, or were they just research scientists?
They were very much research scientists, so they didn't have a well-defined entrepreneurial experience, but I do think that people who set up academic labs can be entrepreneurial in their approach.
Very much so. Oftentimes you have to scour the world for funding or for supplies or collaborators, employees. Yeah, that's probably very similar to running an early stage startup.
It is, and then you have to sell your science. You have to get people, including trainees and funding agencies, excited about what you do. So there are some similarities.
So let's talk a little bit about Richard's work and your work as a postdoc and how that led to your understanding that this could be a startup.
His lab has been studying the extracellular matrix for more than 5 decades. Mm-hmm. And when I came in, there was a lot of information about what is in the matrisomes, which is collectively a composition of matrix proteins in different tissues. So there was a lot of information about what is in the matrisomes of diseased tissues. There had been a really big collaborative project with the Broad Institute where we were characterizing the matrisomes of tumors from patients. When I came in, I was interested in metastases, so I looked at what was in the matrisomes of metastases. And it was very clear that the matrisomes of diseased tissues are very different from the matrisomes of normal tissues. That information existed, But to convert it into a therapeutic opportunity, we went about to build tools to target these proteins. And that's what these nanobody libraries were about.
Can you explain to our audience who might not know what nanobodies are, what they are, how you make them, and why you chose to use them?
Of course. So nanobodies are single domain antibodies that are derived from what are called heavy chain only antibodies found in camelids such as alpacas and llamas. At Matrisome, we chose nanobodies because the matrix is dense and cross-linked, and it's typically viewed as a barrier. It prevents drugs and immune cells from infiltrating and attacking disease cells. And nanobodies are really small. They're about 10 times smaller than a conventional antibody. They're also stable, and we felt that they were the right tools because of their size and stability to penetrate the matrix and bind to it in order to deliver cytotoxic payload. So that is really why we've chosen nanobodies as our basic engineering tool.
Can you explain to us what is the matrix? What does it consist of, or what does a tumor consist of really? And how you're leveraging that to target your specific therapy?
Yes, so as you know, tumors are largely made up of cancer cells, but beyond cancer cells, there's a lot of non-cellular material that tumors, in fact our entire bodies, constitute of a lot of non-cellular material. And a large part of that non-cellular material is called the extracellular matrix, or collectively the matrisome. It's essentially a network of proteins that surrounds or is outside every cell of the body. Normal cells, skin cells, cells of the tissues, and the matrix is also present around disease cells, around cells of tumors and metastases. And this meshwork of proteins, it's a network of hundreds of proteins. They play a role in normal physiology and also in disease progression. Matrix remodeling and deposition is a hallmark of all chronic diseases. That includes tumors, it includes fibrosis, it includes cardiovascular disease. An analogy that I typically give people is like bricks and cement, where bricks are the cells and the cement is the matrix that's around it. And in the context of disease, this matrix gets very abundant and very dysregulated. And now the question is, why are we going after these proteins as opposed to cells? When you just think about oncology and cancer, the typical targets of targeted therapies have been cancer cells. And that makes sense, they are the agents of disease. But cancer cells are heterogeneous. They change, cell surface receptors mutate. Compared to this heterogeneous target, our targets in the matrix are very stable. They're very abundant. They're not subject to mutations, and they're widely expressed, as I mentioned, across solid tumors and also in metastases, and largely absent from normal tissues. So our core philosophy is to convert this matrix from a barrier into a drug depot.
So the targeting of the matrix will allow you to specifically direct the therapy to the tumors, even when the tumor cells might no longer be looking like they used to at the beginning of the cancer.
Absolutely.
Right. Yeah. So do you hope to avoid resistance mechanisms that way?
That is definitely a possibility. I think what our hope is that when we think about targeted therapies, there is a certain amount of therapeutic index that can be very effective. The matrix as a target is very abundant, it's stable. And so when we deliver therapies directly to it, the idea is that via matrix binding, sustained presence of the drug at the disease site, there is targeted delivery proximal to cancer cells or disease cells, and because the targets are selectively expressed, there is only specific activity. It's targeted activity, and there's systemic clearance, so there's less systemic toxicities. All of this combined can really improve therapeutic index. So that's really the hope.
So the Matrisome therapeutic technology is now built on the knowledge of very specific disease-correlated targets.
Yes.
The methodology of targeting them using nanobodies.
Yes.
And then a payload that you're not disclosing yet, right?
Yes. We are, we're very much a payload agnostic technology.
Okay.
And the targets in the matrix that we're prioritizing are also very, very widely expressed across solid tumors. Mm-hmm. What I also want to highlight is, the big unmet need in oncology is metastasis. 90% of cancer-related patients still succumb to this disease, and that's largely because cancer cells are heterogeneous. Mm-hmm. They change, they mutate, and they evolve, and current cancer targeting therapies don't seem to work on advanced stage disease. The hope for Matrisome Bio is that some of our matrix targets are expressed widely across human metastases as well. And so if we're delivering therapeutic payloads to tumor matrix, we're also delivering them to the matrix of metastases. And that's really the hope that we have for the platform.
Fast forward, that could make a promise to have a sort of a primary cancer agnostic therapy.
Yes.
Is that right?
Absolutely. So we are very much a cancer agnostic platform. And the nanobodies can be used to deliver a variety of payloads. It could be small molecules, it could be isotopes, it could be cell therapies. And while we are a platform with the given market and with the limited amount of capital and team that we have, we are staying very focused. We are very much prioritizing a class of payloads. We're very interested, in fact, in radiopharmaceuticals and are really focused on developing first-in-class and best-in-class matrix-targeted radiotherapies.
Now, Matrisome is pursuing these important solutions for cancer patients.
Yes.
And you've taken on the challenge of running this company.
Yes.
As a scientist.
Yes.
Let's talk a little bit about the life of a startup at LabCentral. How many people do you have at Matrisome now?
So we're 5 people, going on to 6. These are 6 full-time people. We also have 3 fractional people, so it's about a team of 10. But when we started out in January of 2023, it was just me. And sort of the internal joke was I was chief only officer because I was the only person on the team. And in terms of my startup journey and going from being a scientist to a founder, it's been something we've been working on for almost 2 years. As you know, Matrisome is an academic spin-out and my co-founders are both academics. Richard Hines has been a leader in the matrix biology space and I was his trainee, but neither one of us had done a company before. Yeah. And when the data from this technology and project started to come out, it was very clear that there was strong potential for translation. And we made a very conscious decision around 2021 to build a company. But since neither one of us had done it, we really leveraged the ecosystem at MIT to figure out what we were going to do next. One of the first things that I had done was I reached out to someone called Kiran Majumdar-Shaw, who is a biotech pioneer based in India, and I used to be a Majumdar-Shaw oncology fellow. That's how I knew her from MIT. And she introduced me to 5 people. One of those 5 people ended up being our new and next co-founder, Sangeeta Bhatia. And so we spent a lot of time talking to people, learning from them. How do you found a company? How should you think about fundraising? How do you think about team building? And these are hundreds of conversations over 2 years. And the most important thing I realized is that I knew very little and I needed the right advice. So when Sangeeta started to work with us, we organically formed a team and we co-founded the company while we were still at MIT. And again, that opened up a lot of experiences for us because Sangeeta is not just a faculty at MIT, she's a serial entrepreneur.
Sangeeta has actually had a number of companies here already, right?
Amazing, yes, she does, yeah. I think Matrisome might be maybe her 6th Oven startup.
Wonderful.
So just having that sort of strategic input, someone who'd done company creation, understood technologies, what was important in the seed stage, was very valuable to us. And that opened the doors to lots of other connections, lots of introductions, lots of advice. And I think how we've really gone about this is taking advice from the best people that we can.
Wonderful. Yeah. A big part of building the startup, other than having mentors and advisors, will be to tell your story to other audiences,
yes,
including investors, because you need money to do this. Have you raised any money for Matrisome so far, and how has that gone? What have you learned along the way?
Yes, we have raised money.
Okay.
We closed our seed round in December of last year in 2023. We are now venture backed, and we're actually backed by 3 venture capital groups and a biotech angel. It has been a really incredible journey for me. As you know, going from being a bench biologist to coming on as in this role of a founder and a CEO, and then really selling the mission and vision of the company. I think in that journey, the most important thing for us was to find investors who were mission aligned, who believed in what we were doing, in how we were going about it. And I think we've been successful. I think we have a very strong investor team that is very invested in the potential of matrix targeted therapies, not just in oncology, but across matrix-related disorders. I think one of the ways that the company really evolved was getting constructive feedback from the hundreds of investors we spoke to. And I am very grateful for that experience because I realized that people really want to help. And almost every conversation, whether the investor invested in us or they didn't, was helpful with the feedback, with the advice, with what they think we should be doing. So it's been an incredible learning curve and a very fruitful experience.
It's great to hear that you've had even beneficial discussions with investors who didn't end up investing in your company. Have you fundraised in Boston or in other places?
So we have fundraised across the US and in fact internationally as well. We've spoken to investors in the US, in Europe, also in India. One of our investors is based in Taipei, for example. And across the board, I think we've gotten a lot of support and interest, despite the markets being tough.
Mm-hmm. Yeah, you said you started thinking about it in 2021.
Yes.
And then you closed your round in December of 23.
Yes.
That's not for the faint of heart.
It is
That's
not.
not been a great period in biotech.
It is not, but in many ways, I think, a lot of people tell me it's a hard time to raise capital. But I think in some ways, being naive and new to the space is an advantage because I didn't have a benchmark. So I didn't know what was hard and what was easy. We just kept going despite the challenges. And we think we're in a good place right now. We think we have the capital and the team to get to our next milestone.
Yeah, let's talk about that. Much of developing biotechs is about reaching meaningful milestones and value inflection points.
Yes.
What are you working on right now, and what do you hope to achieve over the next 6, 12, and 18 months for Matrisome?
So we are, our team is operating here at LabCentral. As I mentioned, when we started in January, there was just me. And since then, we're now 6 FTEs, a lot of people from industry. And Matrisome is right now in preclinical development. So we are testing and doing proof of concepts for our interested class of payloads, radiopharmaceuticals, and really planning our path to the clinic for this first-in-class matrix-targeted therapy. And that is really our next milestone. In the seed stage, we want to essentially come to a development candidate and then raise on that to try and take that asset to the clinic. But imagining success, we'd also like to raise additional money to expand our platform.
Yeah. Are you going after a single candidate for now? A single target with a single payload combination?
Yes. What we are doing in the seed stage is focusing 80% of our capital and effort on that single asset. But we are spending 20% of our time to keep or to further de-risk our platform as well, so that in the next round we not only raise to take this asset to the clinic, but also are ready to expand and pivot to other payloads, other indications, and perhaps even other targets.
Are you so what's the timeline that you're talking about? How long will it take you for candidate selection, and when do you think you could have your first clinical trial starting?
Yeah, we're early. But the current plan is that we're hoping to have a development candidate and an IND filed in 2026 or 2027. Mm-hmm. And so that's about 2.5 to 3 years from now.
Let's talk a little bit about your journey here at LabCentral and what's different from being a lab scientist at LabCentral from being a lab scientist at the Koch Institute.
I think what's really amazing about LabCentral is the innovation ecosystem. So when we chose LabCentral as the incubator to build our company, we were really attracted by the fact that it is a premier biotech incubator. It has amazing infrastructure, it has a lot of good instruments, and there was of course talk about the ecosystem, but I don't think we quite realized what it meant. And now that we've been building the team here, I think being at LabCentral gives us access to a lot of early stage startups. They're all going on similar journeys as us. They're all starting to tackle really difficult questions of biology, address really big unmet needs, and are also figuring things out just like us for the first time. So there's a lot of camaraderie, there's a lot of help, everybody helps each other. For me personally, being at LabCentral has been helpful because it has allowed me to connect with a lot of other first-time founders and first-time CEOs, and also experienced CEOs, because this ecosystem has a combination of all of them.
What are some of the lessons that you've learned from peers that weren't coming from your board?
Well, so many. I think there's a lot of operational challenges every day. There's a lot of decision making that has to be done. We're all on steep learning curves. And I think the biggest thing I feel is we're all operating in an information poor environment. So decisions have to be made with the information at hand. And I think reaching out to other people in the ecosystem who've walked that path or are walking that path with you, and seeing how they go about this, learning from their experiences, that's been incredibly helpful. Which is, I think, it's not something that the board necessarily would get into. So having that ecosystem, I think, is quite helpful for us.
If you were to advise your younger self, maybe an early postdoc at MIT who was thinking about maybe starting a company at what point, what are some of the learnings that Noor has under your belt right now that younger person didn't know yet?
So much. It's so interesting. I was right at the KI down the street, which is 5 minutes away, but it's quite insulated, right? We're in academia. We're thinking about. I had a very different path in mind, which was to be an academic, and it was only when I moved in here, and before this we were at another incubator for a while, that I realized the amount of innovation that was happening. I wish I had the opportunity to connect with that innovation ecosystem while I was still at MIT. It may have helped accelerate how quickly the company came about. It may have made me a bit better prepared for the challenges that we did face. I think that's a big learning for me.
Very good. You know that we love the color orange, and we've asked you to bring an orange object. What did you choose to bring?
So I brought an orange pair of sunglasses right here, because I got these for the Orange Odyssey.
Oh, nice! Yeah, for our 10-year.
For the 10-year celebration of LabCentral.
Excellent,
yes.
excellent. Yes. Good. Well, thank you. Thank you for being available for our interview and sharing your learnings. We wish you luck for the next 4 or 5 or 10 years with Matrisome.
Thank you.
And success for this very important work that you're doing for
Thank
patients.
You so much.
Thank you.
Thank you so much.